This is module 2.2.2 of the online course on genetic selection from concepts to application to genetic improvement of health. My name is Jack Dekkers from Iowa State University, and I will be the presenter of this module, which focuses on Mendelian inheritance and linkage between loci. In this module, we'll discuss Mendel's laws of inheritance. We'll talk about genotype and allele frequencies in populations. We'll talk about Hardy-Weinberg equilibrium, about linkage, map distance, and recombination rates between loci, about haplotypes and haplotype frequencies, and about linkage disequilibrium, which is a very important concept with regard to finding genes or QTL associated with quantitative traits. To target your learning, I'd like you to keep the following questions in mind as we go through this material. First, is Mendel's law of independent assortment correct? Second, how do genotype frequencies at a locus relate to frequencies of the alleles at that locus? Third, how does the recombination rate between loci differ from the map distance between loci in centimorgans? Fourth, what is the recombination rate for loci that are on different chromosomes that are unlinked? Fifth, what are haplotypes? Sixth, how do haplotype frequencies relate to the frequencies of the alleles that make up that haplotype? And finally, what is linkage disequilibrium and why is it important? To begin with, I'd like you to watch the following 3-minute TED Talk that is embedded in this link, which is a simple illustration of Mendelian inheritance. This video illustrates Mendel's first law of inheritance, which is the law of segregation, that states that during gamete formation or meiosis, the two alleles that an individual carries at a gene segregate from each other during meiosis, such that each gamete carries a random one of the two alleles that the parent carries for each gene. This is illustrated here, which shows how pairs of alleles that an individual carries are transmitted to offspring as individual alleles through the germ cells. Then, through fertilization, the allele received from the mother is combined with the allele received from the father to recreate the diploid genotype in the progeny. Mendel's second law of inheritance is the law of independent assortment, which states that genes for different traits segregate independently during the formation of gametes. For example, in the case of the color and smoothness of peas, Mendel found that which of the two alleles for color that a parent passes on to a progeny is independent of the allele for smoothness that parent passes on to its progeny. However, we now know that this is only the case when the genes responsible for these two traits reside on different chromosomes. In other words, when they are unlinked. And in fact, when we use genetic markers to identify genes that are responsible for traits, we actually rely on the dependent assortment of alleles at linked loci or genes. And we'll discuss this later. In addition to genes for different traits, Mendel's law of independent assortment can also apply to two genes that affect the same trait. In fact, it applies to any pair of genes or loci, but again, it is only valid if those loci are not linked, if they are on different chromosomes. The purpose of this slide is to illustrate the terms genotype frequency and allele frequency. Assume we've taken blood samples from 747 people in Iceland, and we've determined the blood group of each person for the MN blood group system. This system is determined by two alleles, M and N. They're codominant, which means that each of the three possible genotypes has its own blood group phenotype. The numbers of individuals observed for each blood group can then be used to compute genotype frequencies, as illustrated here. So the frequency of the MM genotype is simply the fraction of observed individuals that have that genotype. Allele frequencies, sometimes also referred as gene frequencies, can be obtained by simply counting the number of copies of each allele that you see in the sample. So because people with the M blood group carry 2 copies of the M allele, and people with the MN blood group carry 1 copy of the M allele, The frequency of allele M in this population is 2 times 233 plus 1 times 385 divided by the total number of alleles that we have sampled, which is 2 times 747, resulting in a frequency of 0.43 or 43% for the M allele in this population. We can calculate the frequency of the N allele in a similar manner by counting, but we can also simply get it as 1 minus the frequency of the M allele, because there are only 2 possible alleles at this locus, so these frequencies have to add up to 1. One important question to consider for a population is how allele frequencies relate to genotype frequencies in that population. Because genotypes in the progeny are created by combining the allele that is passed on by the father with the allele that is passed on by the mother, the relationship between allele and genotype frequencies depends on how males and females are mated. For most loci, mating can be considered to be random, and there's no reason to expect that males of a particular genotype more likely mate with females of a given genotype at that locus. Exceptions would be genotypes for genes that are associated with traits that are important in mate selection, but these are exceptions rather than the rule. So if mating is at random, then the frequency of a given genotype in the progeny generation can be obtained by simply multiplying the frequencies of the 2 alleles that create that genotype among the sires and the dams. So if mating with regard to the MN blood group is random, and the frequency of the M allele is little p among the parents, then we expect the frequency of the big M genotype among the progeny to be p times p, or p squared. Similarly, the frequency of NN is q times q, or q squared, and the frequency of heterozygotes is p times q plus q times p is 2pq. These are what are called the Hardy-Weinberg equilibrium genotype frequencies, named after Hardy and Weinberg, who simultaneously but independently discovered this relationship between allele frequencies and genotype frequencies in a population under random mating. So under Hardy-Weinberg equilibrium, the frequency of the 2 homozygotes, MM and NN, in the population are p squared and q squared, and the frequency of Mn, the heterozygotes, is 2 times p times q. So for our blood group example, given the allele frequencies of 0.57 and 0.43, we can then use the Hardy-Weinberg equilibrium law to compute the expected genotype frequencies under Hardy-Weinberg equilibrium in the Iceland population, as is illustrated here. So the expected frequency of the MM genotype is 0.57 squared, and that of NN is 0.43 squared, and that of MN is 2 times 0.43 times 0.57. We can then compare these expected frequencies to the observed genotype frequencies in the Iceland population. In this example, the observed genotype frequencies are very close to the expected frequencies, so we can conclude that the population is in Hardy-Weinberg equilibrium for this locus, which implies that mating was at random for this locus at least. We can actually test this statistically by a chi-square test. For details, I'd like to refer you to this Wikipedia link that is provided here. Genotype frequencies under Hardy-Weinberg equilibrium result in the following relationship between allele and genotype frequencies. So the maximum frequency of heterozygotes in a population is 0.5 under random mating, which occurs when the frequencies of both alleles are 0.5, as indicated here. Another important relationship is that for loci for which one of the alleles has a very low frequency, most of the alleles are present in heterozygotes. For example, if the frequency of big M is 0.1, then the frequency of the MM homozygote is 0.1 squared, which is 0.01, and the frequency of heterozygotes is 2 times 0.1 times 0.9, which is 0.18. So if there are 20 M alleles in a population, then 2 of them are expected to occur in an homozygous M individual, and the remaining 18 out of 20 are expected to be in heterozygotes. This is important in case, for example, the M allele is a deleterious recessive allele, such that MM individuals are lethal, but heterozygotes are normal. In that case, only a small proportion of individuals in the population are affected, although the frequency of the deleterious allele can still be substantial, for example 10% as in this example. Thus, although the M allele will be selected against, because homozygous M individuals don't reproduce, it is very difficult to get rid of the allele because most of the M alleles sort of hide in heterozygotes. This is one of the problems of removing genetic defects that are controlled by deleterious recessives from a population, unless you can specifically identify the carriers by, for example, test matings or by genotyping if you have a genetic test. This diagram illustrates the independent segregation of alleles at two unlinked loci. So an individual that is a double heterozygote for these two loci produces 4 gametes. The frequency of each possible gamete is 1/4. The alleles at these two loci segregate independently because a gamete that receives the G allele at the first locus is equally likely to receive the big Q or the little q allele at the second locus. A situation where the two loci are located on the same chromosome, so when they are linked, is illustrated here on the right of the slide. And the combination of alleles at loci that are on the same chromosome of the pair is referred to as a haplotype, with each individual thus carrying 2 haplotypes. And these correspond to the alleles that came from the paternal and the maternal gamete that created this individual. In this example, the individual received the G big Q haplotype from its father and the A little q haplotype from its mother. Note that if the loci are linked, a double heterozygous parent will still produce 4 possible gametes, but their frequencies will not be 1/4, which is what they're expected to be when the two loci are unlinked, as illustrated on the left. Instead, the frequency of gametes that carry one of the parental haplotypes is expected to be larger than the frequency of gametes that are a result of a crossover between paternal and maternal chromosomes. These are considered recombinants. So the non-recombinants are G big Q and A little q gametes, and the recombinant gametes, which result from crossover, as indicated here by a picture out of the original paper by Thomas Morgan Hunt, who developed the idea of crossover or recombination. Those recombinant gametes are G little q and A big Q. So in fact, how closely two loci are linked can be quantified by counting the proportion of recombinants that are produced during meiosis, and that's called the recombination rate. Thus, if the recombination rate is C, then the proportion C of gametes produced are recombinants, 1/2 C of each, and a proportion 1 minus C of gametes are non-recombinants, again half times 1 minus C of each. So if the recombination rate is very low, for example 1%, or C is 0.01, then almost all of the gametes produced are non-recombinants, and they will carry the non-recombined parental haplotypes, and almost none of them are recombinant. Note also that if the loci are unlinked, this must mean that C is equal to 1/2. So unlinked loci have a recombination rate of 1/2, because that results in each of the 4 gametes having a frequency equal to 1/4. So two loci that are on the same chromosome are said to be linked, but depending on their distance, they can be closely linked or almost unlinked. Distance between loci can be measured on several scales. Nowadays, with the availability of sequence data, we can measure distances in base pairs or kilobases, as illustrated here, between genes and also within a gene. This is referred to as physical distance. Now, in the age of cytogenetics, when chromosomes first could be identified under the microscope, distances were measured relative to the numbered G-banding pattern that can be made visible by staining when the chromosomes are condensed during the metaphase of mitosis, so as shown here. Then in the era of linkage genetics, distances between loci were measured by recombination distance or linkage distance, which quantifies the probability of a crossover occurring during meiosis. On this scale, two loci that are 1 morgan apart, on average, have one crossover during meiosis, which is when the maternal and paternal chromosomes of a pair exchange DNA to produce recombinant gametes. It is important to note, however, that this linkage distance is not exactly the same as the recombination rate between loci, because if two crossovers occur between those two loci, then non-recombinant gametes are produced between those two loci, because the two crossovers cancel each other out. So only if the number of crossovers that occur between two loci is uneven will that meiosis result in gametes that are recombinant for those two loci. This makes the recombination rate between two loci to always be less than the map distance between those loci. To give you some idea of scale, the size of most mammalian genomes is around 30 morgans. Now pigs have 19 chromosomes, so an average chromosome is 1.5 morgan. This means that there are on average only 1.5 crossovers on an average pig chromosome during the meiosis. So although recombination rearranges haplotypes, the number of rearrangements that occur on a chromosome is relatively small. Now we can also consider the statistical dependence or independence of alleles that we see in haplotypes in a population. If the alleles that are present at the two loci are independent, then the frequency of a haplotype in a population is simply the product of the frequencies of the alleles that make up that haplotype. So if the frequency of G is 0.3 and the frequency of big Q is 0.6, then under independence of these two loci, the expected frequency of the G big Q haplotype in the population is 0.3 times 0.6 is 0.18. In the same way, expected frequencies of the other three haplotypes under independence can be derived, resulting in haplotype frequencies of 0.12, 0.42, and 0.28. So those are the haplotype frequencies that we would expect if the alleles at the two loci are independent of each other. The observed haplotype frequencies in a population can, however, differ from that expected under independence. For example, consider a population where the observed haplotype frequencies are 0.1, 0.2, 0.5, and 0.2. So in this population, allele G more often goes together with allele Q than expected under independence, and allele A more often goes with allele big Q than expected. If the observed haplotype frequencies differ from those expected under independence, then the loci are said to be in linkage disequilibrium. Linkage disequilibrium is actually a very important concept in the use of genetic markers for selection. For example, assume that the Q locus is a resistance locus and the AG locus is a genetic marker. Now also assume that we can't genotype individuals for the resistance loci, for example, because we don't know where it is, but we can genotype individuals for the marker locus. Then if we know that the A allele at the marker locus is more often associated with the resistance allele big Q than the G allele, then we want to select individuals that carry the A rather than the G allele. In this case, of all the A alleles, 5 out of 7 are associated with the big Q allele, whereas only 1/3 of the G alleles are associated with the big Q allele. However, this selection on alleles at the marker locus will only be effective if the marker locus is in disequilibrium with the resistance locus. If they are in equilibrium, and the A and G allele are equally likely to be associated with the resistance allele, then there will be no effect of selection. So linkage disequilibrium is an important concept, and we'll utilize this multiple times in the upcoming modules. We can quantify linkage disequilibrium between two loci based on a measure called R-square, which is the square of the correlation between alleles at the two loci, as illustrated in this table. This shows the haplotypes that 5 individuals from a population carry. To compute the correlation, we first have to convert the ladder designation of alleles to numbers. For example, and this is arbitrary, we can convert the A allele to 0, and we can call the G allele 1 at the marker locus, and we can call big Q 1 and little q 0 at the resistance locus. Now, once we have these numbers, then we can simply calculate the correlation between these two sets of numbers, which in this case results in a correlation of 0.53. The fact that this correlation is different from 0 indicates that these two loci are not independent, they're correlated. So these two loci are in linkage disequilibrium in this population. Now note that the sign of this correlation is arbitrary because it depends on which allele we set to 0 versus 1. And for this reason we often use the square of the correlation as a measure of LD between two loci, such that the sign does not matter. So here the R square or the LD is 0.53 squared is 0.28. Now R square can range from 0 to 1, with R square equal to 1 referring to loci that are in complete linkage disequilibrium, and R square equal to 0 to loci that are in linkage equilibrium. R square also relates to the R square that we are familiar with in regression analysis, as the proportion of variance that is explained by the model. Along these lines, when you consider the GA locus as an observable genetic marker and the Q locus as the unobserved resistance locus, then if the R square between these two loci is equal to 1, then genotype at the marker completely predicts the genotype at the resistance locus. If the R square between the two loci is 0, then genotype at the marker locus has no predictive ability with regard to genotype at the resistance locus. So for a genetic marker to be useful, it needs to have a substantial R square with the locus that you're interested in selecting for. This figure gives an example of LD in a chromosomal region with multiple genetic markers. The chromosome is shown at the top with the location of the SNPs for which this population has been genotyped. The triangle below shows the LD, the linkage disequilibrium between each pair of SNPs in the squares that connect a pair of SNPs. So for example, this block here shows the LD between this SNP and this SNP. And the color coding, with darker representing higher LD, gives you an overall idea of the LD patterns along this chromosomal segment. And in general, LD tends to be higher between SNPs that are close together, which is represented at the top of the triangle. So the numbers here represent the R squared times 100%. The blocks tend to be lighter at the bottom of the triangle, which represents loci that are further apart from each other. This relationship between the level of linkage disequilibrium and distance can also be represented in the following graph. And in this graph, each point in the plot represents the R square between a pair of SNPs in a population, and this is then plotted against the distance between those two SNPs in kilobases. Although there's quite a bit of randomness, on average LD is higher between SNPs that are closer together and low for SNPs that are far further apart. This makes sense because the recombination rate is higher between SNPs that are far apart, and recombination is what breaks up haplotypes and results in alleles at loci to be more independent. And in contrast, at short distances, there is very little recombination, and therefore LD that is present in the population between SNPs that are close together is not broken up as quickly. However, you should also note that two SNPs that are close together are not guaranteed to be in high LD. There's also a lot of close SNPs that are in very low LD. This is because there's a lot of randomness in both the creation and the erosion of LD, with creation of LD in populations mainly resulting from random sampling of the haplotypes that are passed on from generation to generation, and erosion of LD being due to recombination, which is also a random process. So this concludes this module, and I want to leave you with the questions that I referred to at the beginning of this module. So is Mendel's law of independent assortment always correct? How do genotype frequencies at a locus relate to frequencies of the alleles at that locus? And referring specifically to the case of random mating. Also, how does recombination rate between two loci differ from map distance between loci in centimorgans? And here you think of the crossovers versus recombinants that are created, where recombinants are only created if the number of crossovers is uneven between the two loci. What is the recombination rate for unlinked loci on different chromosomes? What are haplotypes? How are they defined? How do haplotype frequencies relate to the frequencies of the alleles that make up that haplotype, if they are independent, if they are in linkage equilibrium? And what is linkage disequilibrium, and why is it so important? Thank you.